Howdy, folks. Thanks for checking into Mr. Ulrich's Nanobiology.com. I am Mr. Ulrich. In this notescast, we're going to be talking about the chemistry of good old proteins. Kind of important little fellas they are. Why are proteins so important? Well, they come in an incredibly large variety of shapes and sizes because of their structure made up of chains of 20 or so different amino acids folded into different shapes. They are structurally diverse, and that makes them, because they can take on so many shapes, functionally diverse. So they do all of these different things. Certainly, the enzymes catalyzing most of the reactions that are taking place in your body and outside of your body, for that matter. They are what make us animals, holding us together with proteins like collagen and keratin. Certainly, we'll talk about membrane channels later on, but transport across the cell membrane in cell communication and immune function, and just keeping us multicellular organisms moving around, as well as even storing energy in the form of bean seed proteins that we all love when we dig into some peanut butter. Again, what makes proteins so interesting and multifaceted is their structure. They are long chains of amino acids. There are about 20 different amino acids that we need to be concerned with. Being a long chain, that makes them a polymer of repeating units. In this case, the repeating units there are the amino acids. The poly name, as I sometimes refer to them, the name of the polymer then is a polypeptide. There are peptide bonds that hold the amino acids together in the long chain, so that makes it a polypeptide. The side chains then of the amino acids that we'll talk about in a moment can then interact with one another and cause that chain of amino acids to fold back on itself and bond together with other polypeptide chains, making these huge, large, complex molecules that have intricate three-dimensional shapes. Let's look a little bit closer at the structure of amino acids themselves. Each amino acid is going to have a few parts characteristic to each amino acid. It's going to have a central carbon. That central carbon is going to be attached to an amino group or an amine group. That amine group is a nitrogen attached to two hydrogen. That nitrogen, of course, has a pair of unbonded electrons, making it pretty good at hoovering up protons that are floating around, making it a base. On the other side of that central carbon is going to be a carboxyl group, which is going to be pretty good at donating, at losing a proton, making amino acids individually nice little zwitterions, most of them. Also attached to that central carbon is going to be the side group, or the R group, the side chain. This is the variable group. This is the one that's different from one type of amino acid to another. It is the one that gives the amino acid its personality, its individual characteristics. There are non-polar side chains that make the amino acids hydrophobic. There are polar side chains that make the amino acid more polar. And there are amino acids that have acidic side chains. Since it's a zwitterion, this is going to make the amino acid solution acidic. And there are, of course, ones that are basic. Some of the R groups contain sulfur. Cysteine in particular is very important because it's going to form what are called disulfide bridges, which make crosslinks between polypeptide chains or the same polypeptide chain, making it fold back on itself. We'll see these in a moment. On a most basic level, when we start building proteins, we have to start looking at peptide bonds. These are formed through dehydration synthesis, linking the amine of one amino acid to the carboxyl group of another amino acid, resulting in a carbon-nitrogen bond, because we're pulling the OH off of one and the H off of another. And so what we end up with are peptide bonds linking the carbon and nitrogen together in a carbon-nitrogen bond. as the protein is built into a long polypeptide chain, we're going to end up with one side or one end of that polypeptide being a nitrogen or nitrogen-bearing amino end and the carbon-bearing carboxyl end on the other side. Therefore, the repeating sequence of the backbone then is going to be nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. And of course, it only grows in one direction, adding new amino acids onto the carboxyl end of the growing amino acid chain. The structure and the function of proteins are intimately linked. The function depends on the structure. Each protein has its own complex three-dimensional structure. That polypeptide chain gets twisted and folded and coiled into its own unique shape. So each protein has its own unique shape coming from that chain of amino acids. So hemoglobin has its own shape, and pepsin and collagen all get their functionality from the shape that the protein takes on. If the structure and the function of proteins are so intimately linked, then how does a protein get the particular shape that it gets that allows it to have a particular function? How do each one of these 10 different enzymes that are involved in breaking down glucose in order to synthesize ATP, how do all of these different shapes arise? Well, it all comes down to the order of the amino acids that are present in the polypeptide chain. Here's two different views of the enzyme lysozyme. It's an enzyme that's found in tears and other mucus secretions. It breaks down the peptidoglycans, the materials that make up the cell walls of bacteria. So where is this information on how to assemble these polypeptides, these chains of amino acids? How do our bodies, how do our cells know how to put these amino acids together into the right sequences? Well, speaking of the right sequences, what we're talking about here when we're talking about chains of amino acids and the order of those amino acids in the chain is we're talking about the primary structure, just which amino acid comes after which in that long chain. This is determined by another polymer, that is DNA. DNA over here on the right side, as you can see in this view, makes it look much more like polymers. And each one of those side chains that are hanging off, those are the nitrogenous bases, the 4 different nitrogenous bases. So you can see how the linear nature of bases in DNA can correspond to the linear nature of amino acids in proteins. But we're going to talk a lot more about that later on when we get to genetics. Needless to say, slight changes in the amino acid sequence can change the way that those side chains interact with one another, and therefore can change the protein's shape and therefore its function. It only takes one amino acid change, and therefore it only takes one base change in order to change the sequence of amino acids and change the overall shape of the protein and its function. Sickle cell anemia is one such instance where there is a single, in this case, even a single nucleotide that is changed in A for a T. And what that does is it changes the normal glutamic acid in the 6th position, the 6th residue, to change to a valine. Glutamic acid, being acidic, of course, is hydrophilic, and valine with just a hydrogen is hydrophobic, and that does change the way the hemoglobin, the strands of hemoglobin, are shaped. Oddly enough, when it's under normal oxygen conditions, the hemoglobin works pretty much just fine. It's just when it's under low oxygen conditions that the valine, then the hydrophobic region of that valine, can interact with other hydrophobic regions of the chain of the polypeptide, and they stick together and they start forming these long fibers, and that's what causes that sickling shape. The amino acid sequence forms the primary or most basic structure, but we're going to fold these proteins, fold these polypeptide chains up into proteins Uh, when we start talking about neighboring amino acids or amino acids that are pretty close to one another along that polypeptide folding up, we call this local folding. The most common structures that we're going to get from local folding or secondary structures are called alpha helices and beta pleated sheets. The alpha helix forms a twist or a coil in the polypeptide chain. It's formed through hydrogen bonds from the hydrogen of an amine group to the oxygen of a carboxyl group 4 residues away. These are most often represented with the twisted ribbon. Beta pleated sheets are interactions between longer sections of polypeptide chain, and they form kind of like these flat sheets. When we start looking at the different representations, they will include the beta pleated sheets, which in this case are the green arrows, and the alpha helices, which in one I think they're looking like they're red, and the other one looks blue or purple. Um, Again, here's lysozyme. These are 2 different lysozymes down here, or excuse me, they're one lysozyme and one is lactalbumin. These are 2 different proteins, sort of. I mean, they are different, but they're very, very, very similar. And if you look at the shape of them, you can see they have these secondary structures in almost exactly the same spaces. This is an interesting case of evolution. Um, the lactalbumin protein happens in pretty much all mammals, and it's a part of making milk of lactation, where the lysozyme in the egg white, of course, like we said before, is about breaking down the cell membranes of invading bacteria. So here's an example of two proteins that have very similar shapes, but 2 widely different functions. It's kind of cool. Secondary structure involves local folding between neighboring or short chains of the polypeptide, short sections of polypeptide. Tertiary structure, we're talking about gross or whole molecule folding. This is going to be interaction between R groups that are located further apart from one another. There are a bunch of different types of interactions that we get, whereas in secondary structures, they're hydrogen bonds. Here we still get hydrogen bonding, but we also get some hydrophobic interactions. We get disulfide bridges, we get ionic bonds that are forming between them. We get a wide variety of bonding. The quaternary structure involves different polypeptides themselves interacting with one another. Um, Collagen is an important protein, makes up skin and tendons. It's actually made up of 3 different chains that twist together into a triple helix, part of what gives it the strength and resiliency that it has. Hemoglobin is actually 4 different polypeptide chains, 2 alpha and 2 beta in most of the hemoglobin that we need to know about. Um, Without these different polypeptide chains interacting with one another, the ultimate structure can't be achieved, and therefore the ultimate function. So we need to have in order to have hemoglobin working, you have to have both of the chains. In order to have collagen working, you have to have 3 chains together. So to quickly review protein structure, the primary structure is made up of the amino acid sequence itself, or the sequence of peptide bonds. This is going to be determined by the base sequence of DNA. The secondary structure, those are going to be the hydrogen bonds between neighboring or close to neighboring amino acids on the polypeptide chain, resulting in those alpha helices and beta pleated sheets. The tertiary structure, this is going to be the whole molecule or gross molecule folding. This is where we get the hydrophobic interactions between R groups and the disulfide bridges forming between the cysteines. Um, uh, when we start getting into quaternary structure, this is where we start getting into more than one polypeptide chain itself. We get cysteines as well as hydrophilic interactions that are holding these quaternary structures together. This is a good place to introduce the concept of chaperonin proteins. Chaperonin proteins are special proteins that provide kind of a special place for protein folding to take place. See, some proteins need to fold their hydrophobic regions into positions that they wouldn't be able to do within the cell because the cell itself is water-based, and so the protein is going to interact with the inside of the cell. So this protein keeps that, the chaperonin keeps that folding protein in a different environment that allows those hydrophobic side chains to bend in the right spot. And that's going to allow that protein to take on the proper shape that it wouldn't be able to do if it was floating around in water. Well, here's our friend lysozyme again. And you may be saying to yourself, wait a minute, why am I looking at Mr. Ulrich? Whatever, go with it. So here's lysozyme again. This is lysozyme lysozyme Z, no, lysozyme C. And remember, that's the one that has a very, very similar structure to alpha-lactalbumin that you're going to find in chicken eggs, even though it does a wildly different function in chicken eggs than it does in mammals. Well, how do we know what the shape of these two things are? How do we know that the lysozyme shape is so similar to lactalbumin? Well, one way is through X-ray crystallography, where we bombard pure samples of the lysozyme and the lactalbumin with X-rays, and then take pictures of the scattered X-rays, and then based on the way that happens, the scattering takes place, the pattern, we can deduce something about the structure of the substance. We also can figure out the amino acid sequence from these things, and since we know how the side chains can go together, we can kind of figure out what the typical places where you're going to find beta pleated sheets and alpha helices. They'll have very typical sequences of amino acids. Also, we can plug all this information into computers, and computers will think of all this stuff for us and automate it at this point. So it makes it much simpler. We've been talking a lot about taking amino acids and putting them together in a chain, the right sequence of them so that the side chains of those amino acids can interact and cause those things to fold together and make the right shape so that they have the right functions. I mean, heck, we're going to spend 6 or 9 weeks in genetics talking about just that, how to make proteins and how important it is for that process to occur properly. We need to take a couple minutes to talk about how to undo that process, how to break proteins apart. And I'm not talking about going all the way down to the breaking of the peptide bonds where we're going to get individual amino acids before we start doing that. That's what's happening in your stomach. Before we start doing that, I want to start talking about changes that are going to take place before we put it in our stomachs. I'm talking about denaturing proteins, and this is going to involve changing those bonds that are going to cause the tertiary structures and the quaternary structures to take place. So we're going to disrupt that tertiary structure. We can do this pretty easily by changing the pH or the salinity of the solution. This is exactly what we do with cucumbers when we turn them into pickles. Put them in vinegar and brine, and that denatures the proteins of the cucumber and causes them to become pickles. We can also do this through temperature. One of the reasons why we worry about high fevers is because that can cause some of the proteins in your body to denature and lose their functionality. So what happens is once those bonds that are holding the tertiary structure together break, that allows the polypeptide chain to just simply unravel, and it loses its shape and it can't do its job anymore. The scary thing when it comes to high-grade fevers is that once the fever is brought back down again, many of those proteins, though some of them can return back to their normal shape and functionality, many of them do not. So the rule of thumb with denaturation is once once a pickle, never a cucumber, right? You can't reverse denaturation. You can't uncook a fried egg. Once those albumin proteins have been denatured and are no longer clear and are opaque like that, that's it. They're denatured forever. Well, on that happy note, I think we'll leave it there. And thanks again for checking into Mr. Ulrich's Land of Biology.com. In class, we're going to take some of this information, probably make some models of insulin or something, and look a little deeper into protein chemistry and protein structure. If you have any questions or comments, feel free to drop me an email. Go to the webpage if you got here through the YouTube page. Other than that, we'll see you in class, and thanks for checking in.